Invitation. My name is Simone Carradori. I'm an associate professor in medicinal chemistry, and today I would like to deal with a new field of interest in my research, which is the synthesis and design of new chemical scaffolds as carbon monoxide releasing molecules for the management of inflammation, but not only inflammation. In particular, as a general rule, gasotransmitters are defined as small molecules of gas, usually freely permeable to membranes, but whose effects are not directly linked to membrane receptors. They can be produced endogenously or enzymatically, and their production can be modulated by specific conditions. In general, gasotransmitters have a well-defined specific functions also at physiologically relevant concentrations, and their function can be also mimicked by other molecules which can deliver these gasotransmitters instead of the direct administration of these gases. Their cellular effects can be or not mediated by second messengers. In this case, these second messengers can have specific cellular and molecular targets. Gasotransmitters, the most famous ones, are usually nitric oxide, hydrogen sulfide, and carbon monoxide. Also, if ammonia and methane can be considered as new gasotransmitters, despite their biological role is not well defined till now. The main differences among them is the chemical reactivity. For example, nitric oxide and also hydrogen sulfide can, for their chemical characteristics, interact with a lot of proteins and compounds which are present in our body. This is why the use of compounds releasing nitric oxide or hydrogen sulfide can have not only a specific effect, a physiological effect, but also side effects when they interact with unwanted proteins or compounds. Conversely, carbon monoxide has a very selective target, which is the protein containing iron at a specific redox state, so ferrous iron. And this is very important because carbon monoxide or compounds which can release carbon monoxide can finally modulate only specific pathways within our cells. Usually, carbon monoxide was defined as a silent killer because at increasing concentrations in the environment, it can induce a lot of disease till death. So the most important thing is the connection between carbon monoxide as a poisonous element or as a therapeutic agent. Usually, gas transmitters, as those reported in these slides, can have a lot of second messengers which can modulate it, but also they can interact with other structures. For example, they can interact with the soluble guanylate cyclase, for example, or enzymes, but also channels, in order to modify, for example, the concentration within the cells of calcium. And for these reasons, they can be used also as a therapeutic agents. And in particular, carbon monoxide, but also compounds which can release this gasotransmitter, have been proposed, and some clinical trials are now ongoing, but we do not have drugs approved at the moment, towards the modulation of the physiopathological states of our body, in particular regarding the cardiovascular system, so the coagulation, the fibrinolysis, but also inflammation and cancer, and also microbial infections. Carbon monoxide is produced endogenously, as we said, in particular through the catabolism of hemoglobin by the enzyme heme oxygenase. In this case, we can produce carbon monoxide, which usually interacts directly with hemoglobin in order to form a complex. But we can also provide an exogenous administration of carbon monoxide. As such, so in this case, we use the gas by means of inhalation as administration route in order to have a therapeutic agent. The problem is that the administration of gas, in particular carbon monoxide, is not easy. In general, it's not easy to administer a gas as a therapeutic agent, but we have the main problem that we have is that it is strongly bound after administration to our hemoglobin in order to produce carboxy hemoglobin, which is not functional for our body. So the other alternative is to use carbon monoxide releasing molecules, compounds which can be classified based on their chemical composition, for example, in metal-containing CORMs and metal-free CORMs. In the first case, we have a metal which can coordinate several carbon monoxide molecules which can be released in the time. In the second case, we have an organic structure, usually, which can be decomposed or hydrolyzed into several products, byproducts. One of them is carbon monoxide. And in particular, we can also induce this mechanism of release, of carbon monoxide release, using a trigger, which can be the solvent or specific enzymes, or, for example, the radiation with a selected wavelength. In my experience, I worked with this compound, so the cobalt octacarbonyl, which is an orange organometallic compound, which is very known in organic chemistry because it is used as a protecting group, for example, in the synthesis of, in the Nicholas reaction, as a protecting group for alkynes. It is very cheap and it reacts fastly with alkynes in order to generate this kind of complex, so an acetylenic dicobalt hexacarbonyl. And as you can see, there are two cobalt atoms, each of them coordinating 3 molecules of carbon monoxide. Then this is the acetylenic portion, which is then linked to an organic scaffold. This compound is enough stable, but if there is an acceptor of carbon monoxide, it can release 1 molecule after 1 molecule of carbon monoxide. And also, if the mechanism of reaction is not known, it is supposed to be sensible to oxidative environment, and it is not known the chemical product deriving from the release of the carbon monoxide. There are a very masterpiece in 2016 regarding the toxicity of these cobalt-based CORMs, because when we think about metals or ions present in a therapeutic agent, we have to evaluate their toxicity. In this in vivo study, the oral administration rats of cobalt-based CORMs did not induce too many side effects, and in particular, despite a very light toxicity to liver and kidney, but only at higher doses, cobalt was not accumulated but excreted with the urine after consecutive administration. This is important because the molecule deriving from the release of the carbon monoxide, the so-called I-CORM, so the exhausted molecule, can, because of the presence of a metal, have a role in the generation of reacting oxygen species, especially poisonous for mitochondria and also nucleus. But the most important thing is that keeping constant the cobalt reactive, we have to consider also the drug sphere, so the organic scaffold, which is linked to the cobalt compound. Starting from a specific paper of Professor Supuran of the University of Florence, with whom I collaborate, and the importance of these compounds regarding the management of specific inflammation disease, we decided to design novel cobalt-based CORMs, keeping constant the dicobalt hexacarbonyl as the releasing agent, connected through an heteroatom as a linker, which can be oxygen or sulfur, to an organic scaffold. In order to improve the previous work, we decided to introduce different substituted aryls, but also heterocycles, which can be important to have an effect against inflammation, such as acesulfame, so coumarins, pyrazoline, and also benzimidazole. The synthesis is straightforward and usually one pot and very easy. You have to functionalize the main scaffold by introducing an alkyne, usually a terminal alkyne, using propargyl bromide in a basic environment and under nitrogen atmosphere, in order to obtain a propargylated derivative, which can be further functionalized to cobalt complex. In particular, in this case, because acesulfame is a bidentate nucleophile, we can obtain the O substituted and N substituted propargyl derivative, which are both functionalized to cobalt complexes. Coumarins were obtained by the classical Knoevenagel condensation between the proper and 2-hydroxy aromatic benzaldehyde with ethyl benzoyl acetate or ethyl malonate in a basic environment. Then, despite the substitution position 3 of the coumarin, we functionalized the hydroxy group at position 6 or 8 in order to obtain the propargyl derivative, which can be further functionalized to the dicobalt complex. And these compounds are very interesting because especially the position 8 in the coumarin scaffold is the less investigated position, so we can obtain useful information regarding also this position. Regarding instead aryl derivatives, we followed the same synthetic scheme, so the functionalization of the heteroatom, so hydroxy or mercapto, and then the introduction of the cobalt hexacarbonyl in the structure. We obtained different compounds, trying to investigate different substituents, but also enlarging the structure and the complexity, and also using natural compounds derived from plants, such as carbachol and thymol. Regarding benzimidazole, we selected this compound because it was easily obtained by microwave-assisted synthesis and then functionalized, and this compound was previously published by us as a COX inhibitor, so an anti-inflammatory agent. So we would like to see if this substitution could implement or enrich or impact on this biological activity. Regarding pyrazoline derivatives, we selected this compound because we previously synthesized it through the formation of the chalcone and then the ring closure to the pyrazoline ring in order to have monoamine oxidase inhibitors, which are CNS active compounds, and so the involvement of inflammation in the CNS-derived disease. All the compounds after purification and characterization were tested in the well-known and established deoxymyoglobin carbonylation assays at a specific concentration of 20 micromolar. This assay is very easy. It is well known in the literature, but also it is used in the food chemistry market because it is usually used to assess the quality of the meat products. In particular, the assay follows during the time by spectrophotometric analysis the decrease of the band relative to the myoglobin and the increase of the two bands relative to the carboxy myoglobin. So it is important to know not only how much carbon monoxide will be released, but also the kinetics of this release in the frame time. In particular, because CORMs, which are fast carbon monoxide releasers, are usually used as probes, as modulators of ion channels. Whereas a relatively slower carbon monoxide releaser can be administered not only parenterally, but also by the oral route in order to obtain an effect as therapeutic agents, so to have a delayed therapeutic effect. And the acetylenic dicobalt hexacarbon derivatives were shown in the literature to be relatively slower carbon monoxide releaser. These are the compounds that I want to propose today because they are the most interesting regarding the biological activity, and in particular they were tested as well as two reference compounds which are similar, such as the CORM, the first CORM which has been synthesized by Professor Supran group, and also this derivative. So as you can see, the reference compound 5B is a very slow but strong carbon monoxide releaser. Slow because, as you can see, all the compounds can release carbon monoxide after 30 minutes of the assay. Strong because, as you can see, the amount of carbon monoxide released is very, very high. Whereas CORM, the orange one, is a slow and weak carbon monoxide releaser with respect to 5B. These compounds, so the benzimidazole derivative, the pyrazoline derivatives, and also the acesulfame derivatives, are considered as slow and weak carbon monoxide releasers because their curves are overlapped to CORM-based. Regarding the real derivatives, as you can see, they can release more than the previous one. And in particular, it's interesting to note that K4B, which is very similar to the CORM as a reference drug, it just differs from the presence of a bromine atom on the para position of this aromatic ring. And as you can see, this is CORM, the orange one, and this is K4B with the bromine atom. And as you can see, we have an increase, so probably the presence of electron withdrawing group when there is this kind of linker can be beneficial to the release itself. But as you can see, the most important releaser was this derivative with the sulfur atom as a bridge, which can till 300 minutes release more carbon monoxide than the reference 5B. Regarding coumarin's derivatives, they were slow but medium carbon monoxide releasers because their curves are in between 5B and CORM, despite the substitution or the desubstitution at position 3, 6, or 8. These are all the compounds tested, the reference drugs, and as you can see, there is a high variability of the release of carbon monoxide. This is very important because if we keep constant the cobalt hexacarbonyl, the organic scaffold and the linker, the heteroatom, are the only variables which have a strong impact on the releasing abilities of these compounds. So we can modulate the organic scaffold in order to modulate the release of carbon monoxide. In addition, we also demonstrated that also if we change the concentration of the compound in the assay, we obtain similar release of carbon monoxide. Then we tested the compound in a cell model of inflammation. So in particular, rat macrophages, which are very famous to test inflammation also in cell-based assays. First of all the compounds were tested for their viability and so biocompatibility towards this cell line till 48 hours at different concentration, as you can see, from 0, just the control, to 400 micromolar. As you can see from these colored graphs, we cannot observe any kind of decrease which can be statistically significant with respect to the control. So the compounds are biocompatible and safe to be used in this cell line. Then we treated this cell line with an inflammatory stimulus, in this case LPS, at a specific concentration, and then we treated with the CORM, the selected CORM. As you can see in this draft, after a reduction, as you can see in the control 0, a reduction after the inflammatory stimulus, we can have an enhancement of the cell viability when we administer specific concentrations of the selected CORMs, with some compounds more efficacious with respect to the others. For example, think about K1 and K2, which can reach at the higher concentration at least the 100% of viability, but also K7, for example, and just a little bit K8 and so on.